Hi everyone, I hope you are doing well. In today's lecture, we are going to wrap up the discussion about amino acids. In this third part of our series, we are going to learn today how amino acid side chains they contribute to their properties and some examination focus important points where we are going to discuss some commonly found amino acid derivatives, and most of them are non-proteinogenic. That means they don't participate in the protein formation. Okay, so this is important because many competitive examinations they usually ask such amino acids, such unusual amino acids. So pay attention and let's start the discussion today. Now, if we classify the amino acids by R group, and you know that R group is what? It is the side chain of the amino acids. Then we can divide these amino acids into five main classes, and these will be based on the interaction with water. That means their polarity at the biological pH. Okay, so when we are just concerned about how side chains they are interacting with water and how such interaction contribute to the properties of the amino acids, in that scenario we divide these amino acids in five classes. First class is the non-polar aliphatic R groups, and they make hydrophobic amino acids. Now, if you see, the first amino acid is the glycine, and glycine has just H, that means just a hydrogen atom as a side chain. So this is the simplest amino acid, and this could be the simplest structure which could have been possible. So this is the smallest possible R, and therefore it has no contribution to interactions which are driven by the hydrophobic effect, right? Now, good part about this H is because it is so small that it makes this amino acid flexible. Now, because of this particular amino acid glycine, it is likely to find in the regions where proteins need to bend or turn. Remember, when a protein or a polypeptide chain needs flexibility, then glycine is the preferred amino acid. Then comes the alanine, valine, leucine, and isoleucine. If you can see, this is alanine, this is valine, this is leucine, this is isoleucine, and they have a very aliphatic chains. So because of them, they are hydrophobic and they stabilize the protein structure through the hydrophobic effect. Why? Because their side chains have the tendency to cluster together within the protein. So, whenever we need the hydrophobic regions in the polypeptide chain, we can find alanine, valine, leucine, and isoleucine there in numerous numbers. Then methionine. So methionine is the sulfur containing amino acid, and you can see here this is the sulfur containing amino acid, and it is also the first amino acid of the polypeptide chain. All right, so whatever the polypeptide chain is, the first amino acid will be methionine. Proline is again a very unique amino acid because here you can see the side chain, it forms a ring by bonding back to the amino group. So you can see here it is bonding back to the amino group. Okay, and that forms a very rigid secondary amino group structure, and we call it as a imino group. So proline, it gives the rigidity, and because of this it reduces the structural flexibility. Now this functions opposite to the glycine. Now glycine is flexible while proline is rigid, and it reduces the structural flexibility. So in the polypeptide chain, wherever the flexibility is not required, the rigidity is required, the kink is required where the prolines come into the picture. So it is also called as a helix breaker because it disrupts the regular structures of the protein. All right, and it causes kinks or bands in the polypeptide chain. Therefore this proline, it is a a very different amino acid as compared to the other amino acids. Now the second category is the aromatic R groups. Okay, so here we have the tyrosine, tryptophan and phenylalanine. So you can see all have the aromatic groups, right? Now this aromatic group, they are very important because they absorb the UV light at a wavelength of 280 nanometers. So you can see here, here is the tryptophan, here is the tyrosine, and this is the 280 nanometer wavelength, and they are giving the best peaks here somewhere around this region, 280 nanometers, and that is the basis of the characterization of the protein, right? So phenylalanine doesn't take part much here. You can see here its absorption is not that great, but tyrosine and tryptophan, and out of these two, tryptophan has the greatest contribution for the absorption of this particular wavelength. So all can contribute to the hydrophobic effect because of the presence of this aromatic R group. Okay, that. The third category is the polar and uncharged R group. So there is no charge, but they are still polar. And we have serine, threonine, cysteine, asparagine, and glutamine. So asparagine and glutamine, they are the amides of the two other amino acids, aspartate and glutamate. All right. And cysteine is very unique again because this is the only amino acid which forms a disulfide bond with another cysteine molecule. So you can see here, this is the cysteine molecule. Here also we have the cysteine molecule, and they have the thiol group in the chain, and then when these thiol groups make bond, they eliminate here hydrogen, and therefore they make a disulfide bond or bridge, right? Now this disulfide linked residues, they are hydrophobic in nature, okay, and they are very much useful to stabilize the structure. So you can find these disulfide bonds in a number of proteins, for example amino acids, for example insulin, which is an hormone, and there are so many examples there. Okay, serine and threonine, here you can see the polarity is contributed by the hydroxyl groups here. So you can see here, this is serine, this is threonine, and they you have the hydroxyl group, and therefore this hydroxyl group, they gives the polarity. The fourth category is the positively charged basic R groups. So basically they are basic. Why? Because they are positively charged groups, and they are the most hydrophilic R groups with the positive charge. Okay. Now in this category we have lysine, arginine and histidine. So lysine has a second primary amino group at the epsilon position. So you can see here, this is the alpha, beta, gamma, delta and epsilon position, and here you can see the amino group here. Okay, arginine has a positively charged guanidinium group. So you can see here, this is the guanidinium group which is a positively charged, and histidine, it has an aromatic imidazole group. So you can see here, this is the imidazole group here, and this is the only common amino acid which have an ionizable side chain with pKa near pH 7. So that means near biological pH. So it can be ionized, and therefore it can both accept as well as donate the protons easily, right? So that property, it makes it a very useful amino acid for active sites in the enzyme and the proteins which take part in the proton transfer reaction. So you will find histidine in such places. Okay. Now, then we have the negatively charged R groups, and they are the most hydrophilic R groups with negative charge. And we have two examples here. One is aspartate, another is glutamate. So aspartate and glutamate, they both have a second carboxyl group. You can see here, one is here and one is here. All right. So that's how we divide, and we see how these amino acids can behave with water. Now, let's come to the second part of today's discussion, which is about some unusual amino acids, or which are uncommon, or we can say that they are the amino acid derivatives. So they may contain So these amino acids sometimes they are formed by the post synthetic modification. Okay, so post synthetic modifications are those modifications which are done on the amino acids after their synthesis is complete in the protein chain, once they are inside the protein chain. Okay, so we call them as a post synthetic modification. So one first example is the 4-hydroxyproline. As the name is saying that it is a derivative of proline, right? So it has the same cyclic ring. You can see here it has the same cyclic ring as proline, but with an hydroxyl group on the fourth carbon. So this is the fourth carbon, and you can see here the hydroxyl group here, and that makes it more polar than the proline. And you will find this 4-hydroxyproline in plant cell wall and in collagen. Then we have 5-hydroxylysine, which is again, the derivative of the lysine when it is formed by the hydroxylation of lysine at the C5 position. So you can see here, this is the hydroxylation, and it is mainly found in the collagen. All right. Then we have the 6N methyl lysine. Again, it is a lysine derivative, and here one hydrogen is attached to the N6 position, and it is substituted by the methyl group, and you will find it in the myosin. All right. Then we have the gamma carboxyglutamate. It is created by vitamin K dependent carboxylation of glutamic acid. And one of the examples is like in prothrombin, like which are the blood clotting proteins. So you will find this gamma carboxyglutamate in blood clotting proteins. Okay. Desmosine is again a derivative of lysine, and here 4 lysine residues are involved. So you can see here 1, 2, 3, and 4 lysine residues are involved. Now this is very unique to elastin. It is just found in elastin only. All right, and its levels in the body fluids may serve as a biomarkers for alveolar wall injury because of some other reasons or some illness. If some alveolar wall injury is suspected, then the desmosine levels they are determined, and then it is seen, and they correlate with the alveolar wall injury. Then selenocysteine. Now this is the, something uh, very much unique. Why? Because this is considered as the 21st genetically encoded amino acid. So you know that we have the 20 common amino acids which we just seen. So selenocysteine, it contains selenium instead of sulfur in its structure, and it is analogous to cysteine here. So this is co-translationally incorporated. Remember, co-translationally incorporated into proteins, and it makes the catalytic residues in various redox enzyme. All right. And then pyrrolysine, again it is considered as the 22nd genetically encoded amino acid, and it is found in some specific archaea and bacteria which are involved in anaerobic methane production, and it is again co-translationally incorporated into proteins. All right, so you can see here the structure of the pyrrolysine, and this is the structure of the selenocysteine. Then histamine is, I think you all know, it is a very well-known mediator of allergic reactions, and in our immunology lectures we covered a lot about it. So this is the decarboxylation product of the histidine. So this is the histamine, and it is formed by the decarboxylation of the histidine. And then ornithine and citrulline, I mean, this is something you have been hearing since long. They are the key intermediates in arginine biosynthesis and urea cycle. So this is ornithine and this is citrulline. Then we have a set of amino acids like glycine, and then we have gamma aminobutyric acid, or GABA in short, or dopamine. They all serve as a neurotransmitters. All right, and they are very much critical to our nervous system. So you can see here the structure of the GABA, and this is the structure of the dopamine. Now, thyroxine is again, an hormone, and this is very important hormone because that it takes care of our metabolism. This is tyrosine derivative hormone, and it contains iodine, right? So iodine makes it very a very famous hormone, and that is since your 11th or 12th, you know that this is a very important hormone for our body. Although all the hormones are very important, but thyroxine is, it is given more exposure, right? So this is thyroxine here. Then we have the glutathione. So the important, aspect of glutathione is that it is a tripeptide, all right, and it is a tripeptide of the glu, cis, and gly. So in just, comment box, tell me uh, what are the amino acids we are referring to. So this is the gamma carboxylate group of the glutamate side chain, and it forms an isopeptide bond with the amino group of the cysteine residue. Now, what is an isopeptide bond? You have heard about the peptide bonds. Peptide bonds are the bonds which are formed between the one amino group of one amino acid and a carboxylic group of another amino acid. So that makes the peptide bond, and it is a condensation reaction. But in this case, when side chain is also involved in making the peptide bond, then we call it as a isopeptide bond, right? Right, you can see it here, it is found here. Now, when two glutathione tripeptides, they undergo oxidation of their thiol groups, they form a dimeric disulfide linked structure, and this structure is known as glutathione disulfide, which is GSSG. So you can see here, which is, bound by a disulfide bridge here. Okay, so glutathione is very much, taken as a supplement also, because it is considered as to neutralize the toxins, particularly, those which are involved by the oxygen derivatives. Okay, so this is a supplement. Thyroxine is also, nowadays available as a medicated supplement, medicine, particularly when people who have hypothyroidism, so they are given this thyroxine, right? So this were the discussion about, these uncommon amino acids. I hope, you enjoyed this, but you learn them for your competitive examination. They are important, and we are closing our discussion today, and we will see you in the next class. Till then, you take care.